And this slide summarizes now how the multifactorial mechanism in producing thrombosis in heat so are working together with many different factors involved. On this slide you can see the vessel wall here with the pathological site, and you can imagine that at the pathological site with inflammation or blood activation we will have much more platelets activate with release of PF4. If there is inflammation, we will have also release of IL-8, and when platelets are activated, the neutrophil activating peptide 2, which becomes beta thromboglobulin when cleaved by cathepsin G, can be found. You know, we have this kind of chemokines. PF4, IL-8, and beta thromboglobulin are chemokines which are released and which are at much higher concentration than in normal blood circulation. If the patient receives heparin, then heparin will form complexes with PF4, with IL-8 also, with a lower affinity, with beta TG2. And you know, the generation of PF4 and heparin complexes at the pathological site will be different than in blood circulation because the amount of PF4 is much higher. And then the chances to form complexes are higher at the vicinity of the pathological site than in blood circulation. And if the stoichiometric concentrations are met, then complexes will form, and PF4 heparin complexes will bind on endocellular cells, but mainly on platelets, and also on monocytes and leukocytes, and this will target the immune response. Antibodies then will bind to platelets, but also to endocellular cells and to monocytes, and start the pathological process, which will be enhanced by other complementary factors. For example, binding of PF4, heparin, and antibodies on endocellular cells will activate endocellular cell surface and release tissue factor, which will contribute to thrombogenicity. Activate platelets will aggregate together, and this will start also the activation process, thrombocytopenia, and lead to thrombosis. Platelets are activate, not disrupt in this case. And platelets will also bind with monocytes and leukocytes to form aggregates. And a typical pattern which is observed in heat is a high concentration of circulating platelet, leukocytes, or monocytes aggregates. This has been identified in the early 2000, and now it is a factor which is accepted as contributing to pathogenicity of heparin-dependent antibodies. And you can see here the worsening process where the aggregates form. There is a loop that will enhance the pathological process and lead to thrombosis.
Is it typical to see those aggregates like on a blood smear?
Okay, so
is it possible or typical?
So Jonas, can you?
Sure. Can you see the aggregates in the blood smear?
The
aggregates?
Can you see aggregate in the blood?
The aggregate? Yes. The first paper who reported this aggregate was in 2000 by a French group, Bachelot, working with so in Brousset Hospital in Paris, and they show that that aggregates are circulating, and now many groups are reporting the presence of these aggregates.
This has to be considered a blood smear.
It's under the microscope.
The microscope.
Microscope. Est-ce que tu veux voir sur la formule
I cannot answer this question. You know, I don't know if you can see it on microscope, but you know, by flow cytometry you can see the interactions. By microscope also you can see some aggregates. How it interferes now in the blood cell count, I don't know.
From my experience, I work a lot of time in hematology, and we can see aggregate in the under microscopy, but we cannot define really if it is it or other consequences. You know, according to the punction venous, you can have aggregate also, and we cannot really define which is the cause. On the other side, for the biopsy, we can find different thrombus. We can explain the difference of the thrombus according to the biopsy when we preleve the thrombus on the hit patient.
So you got the white clot or the red clot?
Yes. The white clot or the red clot.
Yeah. One is more venous, the other is more thrombosis.
Yeah. And when the monocyte is more involved, the clot is more white than red. And then the platelet is involved, the clot is more red.
So what I can tell you also is then when heparin-dependent antibodies prepared from patients with HIT are mixed with normal blood, then it generates aggregates. And if antibodies, control antibodies from healthy individuals are mixed with healthy blood cells, nothing occurs. There is another way, and this can be observed by flow cytometry, but it can also be observed by microscope. And now there have been many reports showing presence of these aggregates. So what is important to remember from this mechanism is that pathological sites contribute to pathogenicity of HIT antibodies, because the risk to have generation of PF4 and heparin complexes is higher at these pathological sites. This is a very important point. And when you look at clinical reports, especially from work hunting or from other groups, so they tend to say that disease develop at sites of pre-existing disease. It is true for it. It is true also for the antiphospholipid syndrome. So it is another presentation extract from the HIT book concerning the interaction between PF4 and tetramer with the molecular structure, where you can bind, you can see, you know, the lysine ring, lysine and arginine rings, exposing that electropositive charge ring, which is able to bind heparin. And here you can see the different parts of the PF4 with the lysine and the arginine, and these electric charge are exposed like this on PF4. So this slide, you know, focus more on the interactions of antibodies with endocellular cells, and they can release tissue factor, which was first identified, I think, at the beginning of 90s in Philadelphia by the group of Mortimer Pons and Sainz, Arepali, Sainz and Pons, working in Philadelphia Children's Hospital, and they have also contributed a lot in understanding HIT. And here you can see the resting endocellular cell, and when heparin PF4 complexes bind on endocellular cells and bind antibodies, then there is this amplification loop which takes place on endocellular cells, and endocellular cells becomes activate and contributes to pathogenicity. So, you know, the mechanism is not only one. There is contribution of endocellular cells contribution of aggregates, contribution of aggregate platelets, contribution of tissue factor, and all these factors, all together will generate thrombin. And if the amount of thrombin generated exceeds the capacity of blood control of the thrombin generation, then thrombosis will occur despite presence of heparin, which in this case contributes as a trigger for developing disease.